This video is going to get you through Lab 5, which is movement across membranes. It starts on page 33 in the lab manual. It starts out by giving you your introduction and your objectives towards the top of that page. This lab is essentially all about how do different nutrients and molecules move around inside of your body. One of the barriers that we talked about in the cell chapter was your cells are all surrounded by a plasma membrane that is selectively permeable. It will allow some things to pass through, but not other things. So we're going to be talking a little bit about how that's going to work out for us in terms of moving things around inside of your body. So flipping over to the next page and moving down towards the bottom after materials where it says D procedures. First thing that we're supposed to do is talk about Brownian movement. It says to describe what you see in the space below, and so this is what a slide would look like if you put some India ink on it. And what I want you to notice is everything's just kind of wiggling around. It's not moving towards something or away from something, but there is some inherent movement in the particles inside of you. That holds true for nutrients and wastes and just all of the chemicals that you have inside of you. All of those chemicals have movement on their own, and that movement is going to help set up the processes of diffusion and osmosis and filtration and some of the other things that we're going to talk about in this chapter. So your book describes Brownian movement or Brownian motion as erratic, which again means non-directional is just kind of random wiggliness, and non-directional, we're not going towards or away from something, we're just kind of moving around in general. After that, diffusion is the movement of a solute from an area of high concentration to low concentration. A solute is something that is dissolved in a solution, and so it can be, again, nutrients, wastes, respiratory gases like oxygen and carbon dioxide. Those are all solutes that are inside of your body. Well, the smaller a molecule is, the faster it's able to move. So in the setup that we usually do in class, what we do is we take some potassium permanganate and some methylene blue. Potassium permanganate is this brownish colored ring that we have going on right here, and then this is the methylene blue. We put one little particle of each of those in different places at the same time, and then what happens is the potassium permanganate diffuses through the agar faster, and so it ends up generating this larger circle because it traveled farther in a shorter period of time. That's kind of the definition of faster. Methylene blue being a larger molecule, it still travels, it just travels a lot more slowly than the potassium permanganate does. Now, unfortunately, they gave you the line to record your results at the top of the next page without any room to write that stuff down, but essentially what you need to make sure you write down is what I have right here. Smaller particles or smaller molecules are going to move faster than larger molecules are going to. That means something like an oxygen molecule, which is very small, it's just two oxygen atoms bonded together, is going to travel a lot faster than a starch molecule that is much, much, much larger. All right, next up, temperature. Now in lab, if we want things to move faster, we can heat them up. You do this sort of stuff every day if you brew your own tea or you brew your own coffee. If you make your water hotter, it's going to result in tea or coffee that much faster. Now, your body can do this to a limited degree. If you get a fever, things move around faster inside of you. That helps your immune system work a little bit better. But you can't really get yourself too cold or too hot because both of those two extremes are not good for your body as a whole. It can really mess around with the proteins that you have. But as a general rule, hotter is faster. The story that I usually give as I'm talking about this one is there's a new trend where everybody loves cold brew coffee. It tends to have a little bit more caffeine in it than hot brewed coffee or regular coffee, if you will. But if you want to make cold brew coffee, the fastest I have ever seen it made is 30 minutes in a special little brewer. You can make hot coffee in less than 2 minutes, and so hotter just makes things that much faster. Side note, if you're going to make cold brew coffee in like a French press like this, you usually have to let it sit at least overnight in your refrigerator. It just takes that much longer to get your coffee to a decent strength if you're going to make a cold brew. So usually takes hours, not just minutes. Next up. This is something that we usually have the students do, and of course you guys don't have dialysis bagging at home, and so you can't do this for yourself, but this goes back to that whole semi-permeable membrane aspect. What we usually do is we tie off a dialysis bag and then we fill that bag with a starch suspension. It's just cornstarch mixed with water. Now I mentioned earlier starch is a huge molecule. Because it is so large, it is not permeable through a plasma membrane, which means when you put it in the bag, it's not going to be able to escape that bag. Water, on the other hand, is a tiny molecule and it can diffuse through that bag. So what water is going to want to do is diffuse into the bag to reduce the concentration of the starch inside of that bag. So normally what we would do is make the bag and then weigh the bag. And so what we're going to do, under number 4 on page 35, that first blank, it says, what is the weight right after you make the bag? We're going to say 7.2 grams. It's a weight that I have gotten before in the lab, and so just go ahead and fill in your blank with 7.2 grams. What we would do then is we would let that sit for 30 minutes to an hour, and then we would continue on through this, but since we're not actually doing this together, we're just going to go ahead and move to, it's been 30 minutes, yay! We would reweigh the bag after pulling it out of a beaker of water. So now, skip down just a little bit, and it says, what is the weight again? This time our bag is going to weigh 7.8 grams. It's gained weight. The next two questions help to explain why that happened. Has osmosis occurred? Yes. Water moved into the bag to try to dilute that starch. How do we know that it happened? The bag gained weight. And the only way the bag could gain weight is if something were to have entered the bag and the only thing that it had around it was water. So osmosis is essentially defined as the diffusion of water through a semipermeable membrane, and that's what happened here. So yes, osmosis occurred. We know because the bag gained weight. Now, the next thing that we usually do here is try to figure out, did the starch leave the bag? So what we would do is we would take some of the starch suspension from inside the bag, put it into a test tube, and then add some iodine. Iodine is what's known as an indicator solution. It turns black in the presence of starch. So let's see, it says pour the water from the beaker into test tube 1 and add the iodine. What is the test? This is a positive test because iodine turns black in the presence of starch, and that's exactly what happened in this test tube over here. Oh, I did that backwards. Pour the water from the beaker into test tube 1 was what that first question said. Okay, our beaker had water in it. What we wanted to know is, did any starch from the bag get into the beaker? And so if we pour the water into this test tube and add iodine to it, this is what we get. It's just an amber color. The iodine did not turn black. This is just a shadow right here. That's not actually black iodine. This is what a black test looks like. And so no, the starch is not able to leave the bag. It's too large a molecule and it can't fit through the little pores that are in the dialysis bag. So, sorry, I read these questions out of order, but for right under the how do you know, and it says use the same test tubes as before, pour the water from the beaker and add the iodine to it. What is the test? It's negative. The starch could not leave the bag. Then it says to cut open the bag and pour the starch contents into test tube 2. This is the result right here. So yes, the starch is in the bag. Did starch diffuse from the bag into the beaker? No, because we did not test, or we did not get a positive result for starch in the beaker, so the starch could not leave the bag. For the why or why not, again, it's too large a molecule, it can't fit through there. The analogy that I usually like to give you here is, you and everybody in your family fit through your door, and you don't have any problem getting in and out of your house because you fit through your door. However, if you decided to get a pet elephant, would the elephant fit through the door? Well, you and your family members are like oxygen or water. You're small enough molecules that you can pass through the door no problem. But the elephant is like a starch molecule. It's so huge it can't fit through that door without damaging your house in the process anyway. And so that's essentially what happened here. It's just too large of a molecule. Okay, next up, we have this concept of tonicity that we're going to be talking about. Tonicity is about concentration differences between two places. Now, what we need to realize here is that all cells, hold on, I forgot to press a button. Y'all are going to get so tired of me not pressing buttons, but here we go. Okay, so all cells have a set number of solutes that are inside of them. I mean, they've got their nucleus, but they also have oxygen inside of them, and they have glucose, which is a little bit of a larger molecule inside of them, and they've got waste, and they've got all these things. So there's just a set concentration of various solutes that are present inside of that cell. When those cells are in your body, they are surrounded by extracellular fluid, which is also called interstitial fluid, although there's a slight difference between those two terms. That fluid should be what's known as isotonic to your cells. And so here's what we're going to doodle. We're going to say we're going to take one of your cells out of your body, and we're going to put it into a beaker. Now your cell has the set number of solutes present inside of it. The fluid that is outside of your cells is usually what's known as isotonic. Iso means the same, so that means there's the same number of solutes outside the cell as there is inside. If you put a cell into an isotonic environment, that means there's no net movement of water. The cell is not going to absorb water, it's not going to lose water, it's already in equilibrium with the environment, and so there's no change to the cell. This little triangle is delta, that means change, and so I'm abbreviating that. Now, since isotonic means it's the same concentration, hopefully you understand there's two other options, too high or too low. We'll start over here on this side with the too low option. It's called hypotonic, hypo meaning too low. We'll put the cell into a beaker again. The cell's going to have the set number of solutes that are present inside of it, but a hypotonic solution will have fewer solutes in it, in this case, than the cell actually has. So what happens here is water always wants to move into the more concentrated solution. Concentration is dictated by the solutes present inside, so those little spots that I was putting in there. So since there's more spots inside the cell, that means it's more concentrated, so water is going to flow from outside the cell to inside. That is going to cause the cell to swell. So there's different options in terms of what can happen depending on whether the cell is an animal cell or a plant cell, or a prokaryotic cell for that matter. If the cell has a cell wall, the contents of the cell will swell to press against the cell wall, and it will create what's known as turgor pressure. And that turgor pressure helps to keep, again, the cell upright. That is going to be very important for plants maintaining their rigidity so that they can stand up against gravity. So turgor pressure is created when you water a plant with regular water, which is hypotonic to the plant, so the cells in the plant can be turgid or firm and the plant can actually stand upright. The problem in you, however, is that you don't have a cell wall. You just have a plasma membrane, which is a liquid membrane, but it's made out of fats. If your cells start to absorb too much water, it will actually just pop your plasma membrane, and so it can actually cause a human cell to burst, which is not good for you, of course. You want your cells to preferably not burst. All right, next option, hypertonic. Hyper means above or excessive. So in this case, we're going to be more concentrated. So we've got our beaker, we've got our cell, it has some solutes inside of it, but this time there are even more solutes outside of the cell than inside. Now, since water always wants to go into the more concentrated place, what's going to happen is the water inside the cell is going to leave the cell and the cell is going to shrink. Now, when that happens in an animal cell, your cell ends up looking sort of raisiny. That's called crenation when that happens. But again, plant cells are different. They have a cell wall, and that cell wall doesn't move. But the contents inside of the cell shrivel up away from the cell wall. That's called plasmolysis when that happens in a plant cell. So the effects are different, and it all depends on whether or not the cell has a cell wall present or not. So what I usually like to mention as we talk about these things right here is things that in theory people are going to know, like should you drink ocean water? Hopefully you all know no matter how thirsty you get in the ocean, you should not drink the water that's in the ocean. Well, that's because ocean water is hypertonic to the cells in your body, especially in your GI tract. So what would happen is all the cells that line your GI tract are all going to lose their water. The problem with that is that gives you horrible diarrhea when you are already dehydrated, and that's what ends up killing you if you drink ocean water. So the ocean is hypertonic to you. So if you're really dehydrated and you need to take a drink, what you should do is drink a hypotonic solution, aka water, and then what will happen is the water that you drink will absorb into your intestinal cells, and then it can get into your bloodstream and it can rehydrate all the other cells that are in your body exactly like it's supposed to. Now, all of that was related to your GI tract, which that's how you ordinarily consume different liquids. If you are so dehydrated that you've lost consciousness, you can't drink anymore without the risk of aspirating, which essentially is drowning on fluids that they give you. So they would hook you up to an IV. Now, if they hook you up to an IV, that means they are pumping something directly into your bloodstream. If they pumped hypertonic stuff into your bloodstream, all of your red blood cells would shrivel up and die. If they pumped hypotonic solution into your bloodstream, all your red blood cells would burst, and then you would be poisoned by the iron in those red blood cells. That's not good either. So what they would do is they would hook you up to an IV of normal saline. It's isotonic to your red blood cells, but it still gets additional fluid inside of your body. So tonicity is different in different places of your body depending on the needs that your body has at any given time. You are more than welcome to drink hypotonic solutions because that will rehydrate you through the GI system. You just can't put a hypotonic solution into an IV because then you're going to be damaging the red blood cells. They're not the same as GI tract cells. So, next up. There's a little line that they gave you where I kind of want you to label what happens to a plant cell when you water it with the different solutions. So this is Alodia. This is the same type of cells that we kind of talked about briefly in the cell chapter. Notice all the chloroplasts here are jammed off to the side of the cell. That's because when you water a plant with tap water, which is hypotonic to a cell, the central vacuole swells up and it shoves everything in the cell up against the cell wall, creating that turgor pressure that we talked about earlier. If, however, you were to water a plant with Gatorade or a salty water type of thing, what would happen is because that is hypertonic to the cell would lose water, the central vacuole would shrink, and the contents of the cell would shrivel away from the cell wall. So you can still see the cell walls out here. The plasma membrane, though, is right here. So this is not ideal. This would end up killing the plant if it did not get some fresh water eventually. So that's what happens to plants, and it's the difference is because they have a cell wall, you didn't. Okay. After that, we have detection of electrolytes. We usually have this little setup in the lab where we've got light bulbs that have some special electrodes on them, and then we put various solutions under the light bulbs. I don't have that at home, and so I can't show you what would end up happening with those things, and so we're just going to write down the results that we ordinarily get for this. If you put one of those light bulbs into distilled water actually is not a good conductor of electricity, and so neither of the light bulbs, not the small one, not the bright one, would light up in distilled water. This would be like not tap water, but this would be deionized water, or in some cases spring water. It just kind of depends, but this is just H2O, nothing else is dissolved in it. Tap water, which is more similar to lake water, or in some cases even ocean water, since climate change is bringing salt water up into different places, but I guess we won't get into that right now. But tap water usually has some stuff dissolved in it. I don't know where Pantego is, but this is the instructor that made this. She put these up here, but Fort Worth water comes from the Trinity Aquifer, and that aquifer is lined in limestone, and limestone, it's an ion. So if you take tap water from the city of Fort Worth, usually what happens is the small light bulb lights up, so we would record that as dim light, but the big light bulb does not light up, and so we don't get bright light out of that. So more than likely you have heard at some point, if there's a lightning storm, don't get in the pool. Pool is more like tap water. Pools are either saltwater pools or they are chlorinated pools, and chlorine is an ion, and so both of those two things can conduct water. But if there were such a thing as a pure water lake out there, you would actually be safe swimming in it because pure water does not conduct electricity. There's just no such thing as a lake that's just pure water. There's always stuff dissolved in lake water. Okay, from there, we take some of that starch suspension that we had put into a beaker earlier, and then we put it up to the lights. Since starch and the next one we're going to talk about, sugar, these are both made out of covalent bonds. Those bonds are very strong and tightly held together, so they don't conduct electricity. So neither starch nor glucose or dextrose can actually conduct a current, so none of the light bulbs light up for that. Then we get three things that are all ionic. Salt is sodium chloride. Sodium forms a positive ion, chlorine forms a negative ion. When you mix those two things in water, they float around as free ions, and so you get bright light out of those. Because remember, ions have an electrical charge. Electrical charge means can conduct electricity. Acids. We'll take hydrochloric acid as an example. When you put hydrochloric acid in water, it separates into a hydrogen ion, which is positive, and a chlorine ion, which is negative. So again, bright light. Bases. I'll use sodium hydroxide as an example. Again, a sodium ion is a positive ion, a hydroxide ion is a negative ion, so when they separate, once again, you get a nice bright light out of that. All right. After that, I'm giving you some of the definitions that I don't think your lab manual really goes into very well. Dialysis. What you need to understand about dialysis is you have to have a membrane or like a plasma membrane in order for things to pass through it. So things are moving through a membrane in the process of dialysis. Diffusion doesn't have to have a membrane. It can be there, but it doesn't really have to be. Diffusion is just movement from high to low. Filtration. This time we're going to have a filter. Filters are very similar to membranes, but they're not quite the same thing. And filtration is going to rely on gravity. And so if you think about a standard drip coffee maker, you put a filter in it, you put the coffee in it, water comes in from the top and then falls through by gravity, taking the coffee flavoring off with it. And then osmosis also requires a membrane, but it's a diffusion of water through that membrane. So hopefully that's going to help you answer the questions that we have going on page 37. Okay, water flowing over a dam is none of these things, and so E is the correct answer. Cells bursting in distilled water, that would be because you put a cell into a hypotonic environment, so that's osmosis, so D. Meat being dried and preserved with salt, well, salt is super hypertonic to a cell, so that's once again osmosis for D. Perfume reaching the nose of one person. So ordinarily they have this little can of air freshener and they want us to spray the air freshener in one corner of the room, and eventually it makes it all the way to the other side of the room. That's just diffusion, because the perfume or the air freshener that's in there, that's just solute that you're spraying into the air, and eventually it gets from one place to another just relying on diffusion. So diffusion is B. Urea diffusing from blood into water. Okay. What you need to understand about that is ordinarily if things are going to be going from blood into something, they have to pass through the lining of a blood vessel, and so that's dialysis because we're passing through a membrane. Milk at a dairy being poured through a cotton pad. Cotton pad, that's a filter, so that's filtration for C. Water forced from blood into tissues by blood pressure. Once again, that's we're going through a membrane, so dialysis. Exchange of oxygen and carbon dioxide between blood and air sacs in the lungs. That is diffusion. You could have an argument there about it being dialysis because we are passing through membranes to get there, but usually we just call that diffusion, so B. Sugar cube sweetening tea with no stirring, that's just diffusion for B. All right. After that, number 8, fill in the following blanks. Name given to the movement of particles due to molecular motion. That was Brownian motion or Brownian movement. Diffusion, osmosis, dialysis, and filtration are all examples of physical processes, because you don't need a body to do that. They just happen on their very own. Just remember that starch thing that we did with the dialysis bag, that's physical. Which term is most inclusive between those three? It's diffusion, because both osmosis and dialysis are types of diffusion. Number 4, as osmosis occurs in the body, what substance is crossing a selectively permeable membrane? Osmosis is defined as the diffusion of water, so water. Number 5, when other factors are held constant, which substance, glucose or salt, should diffuse faster? This is a question that goes back to size matters when things are going to move. Glucose weighs 180 AMU. Salt weighs considerably less than that. And so the salt is a smaller molecule. It would diffuse faster. There's also a bonding type of thing. The salt is going to split and become even smaller, so just salt is the answer to that. Number 6, in osmosis, net movement of water is into a hypertonic solution. Remember, we always want to go into the more concentrated area, so hypertonic. Next page, number 7, what material is used to test for starch? That's iodine. Number 8, the membranous bag used to demonstrate dialysis swells and may even burst open. What is the obvious explanation in one word? Osmosis. Remember that water moved into the bag to try to dilute that starch out. Number 9, which of these processes is not a special type of diffusion? That would be filtration, since that relies on gravity and not particle movement. Number 10, what term is used for substances that ionize in water? That would be electrolytes, which is actually right down on the next question. What 3 types of compounds are electrolytes? Salts, acids, and bases. So that takes us through everything you needed to know for movement across membranes. Good luck with the quiz.